This video is about repressible and inducible operons. We'll start with a repressible operon. An example of a repressible operon is the trip operon. The trip operon starts on and is turned off. The trip operon is an example of an anabolic pathway. Most repressible operons are anabolic pathways, which means we're building something. This right here is the operon. The operon consists of the promoter region, which is where the RNA polymerase binds, the operator, which is where a repressor protein can bind, and all of these genes right here. Each of these letters represents a gene. E, D, C, B, A, these all represent genes, and these genes produce enzymes that help us make tryptophan. These genes together are known as a transcription unit. Because they're in a prokaryote, they're all transcribed at the same time. Okay? And in this transcription unit, the mRNA again is used to produce proteins that help produce the amino acid tryptophan. So, what is the default setting? The default setting here is the operon is on. What does that mean? That means the repressor protein is inactive, which means the tryptophan producing genes are active and the enzymes to build the amino acid tryptophan are expressed. So in this default setting right here, okay, RNA polymerase can bind to the promoter. It can move down this piece of DNA, because remember, this whole section right here is the DNA. Right? It can move down this section of the DNA. It can produce the mRNA of all these genes. And then we can have translation occurring and we can produce the proteins. These are enzymes that produce tryptophan. So the default setting is that this operon is on, which means a precursor molecule goes in, it goes through these enzymes, and eventually we produce tryptophan, okay, which is an amino acid. So that's the default setting. But what happens when you have enough tryptophan? Well, when there's enough tryptophan present, okay, this tryptophan fits into the repressor protein and it activates it. Okay? The repressor protein can now bind with the operator and stops gene expression, which means it turns the operon off. Tryptophan then is often called the CoRepressor because this helps this process occur. So let me show you this on a picture. In a different location on the DNA, separate from the operon, there is this regulatory gene. This regulatory gene codes for a repressor protein. However, the default setting for the repressor protein is inactive, as we said earlier. Now, when you have enough tryptophan, all that extra tryptophan can't be used to make proteins, because remember, tryptophan's an amino acid, so this extra tryptophan binds with the repressor protein and activates it. That's why tryptophan here is called the CoRepressor. And this is the active repressor protein. This active repressor protein can fit into the operator now it can block RNA polymerase and stop transcription from occurring. That's how we turn the operon on. So this is an example of a repressible operon. We start with it on, meaning we make the enzymes that can make tryptophan, so we're producing a lot of tryptophan. When we have enough tryptophan, remember this is feedback inhibition, this end product, okay, actually activates the repressor protein, which attaches to the DNA at the operator so that RNA polymerase can't bind, shutting down and turning off this operon so that these enzymes can't be made, so that no more tryptophan can be made. Eventually, again, when we run out of tryptophan, the tryptophan will pop out of this repressor protein, causing the repressor protein to inactivate, turning this operon back on. So that's an example of a repressible operon. Let's look at an example of an inducible operon. Our example of an inducible operon that we're going to use is the lac operon. The lac operon is a catabolic pathway, which means we're breaking something down. This inducible operons start off, which means no protein synthesis, no gene expression, and is turned on, which means yes, protein synthesis. Okay? So let's look at our lac operon here. In our lac operon, the operon is set up the same way as in the repressible operon, right? This right here is the operon. We have the promoter. Oops. We have this next part, which is supposed to be the operator. We have the operator. And then here's our transcription unit. These are the genes that are going to be used to produce proteins. Okay, and again, remember, this whole thing right here is our DNA. Okay, so what happens here? Like I said, the default setting is the operon is off. What does that mean? This repressor protein is actually made in its active form. Okay, so here's a repressor protein. It's made in its active form. So default is an active repressor protein, which means it fits into this operator, and that's its default. So that means the operon's off. RNA polymerase cannot attach, okay, which means these genes cannot be transcribed and translated. So when I say the operon is off, we have an active repressor protein. Because it fits into the operator and stops protein synthesis, stops gene expression. However, what happens if you make some sort of sugar? Like let's say you make or you eat lactose and you want to break lactose down. So in a bacteria, okay, a bacteria would not need the enzymes to break down lactose unless lactose was present. So let's say this little bacteria eats some lactose. Okay, so here's our lactose. Remember, lactose is a disaccharide. Okay, so our little bacteria eats lactose, which is a disaccharide. Okay. So we now need to make the enzymes to break lactose down. Okay, so what happens? Well, what happens is we turn the operon on. This sugar, this lactose, it's actually called allolactose. It's an isomer of lactose. Okay. Allolactose acts as the inducer, inactivating the repressor protein. So allolactose, which is a sugar, acts as an inducer because it binds to the repressor protein, which inactivates the repressor protein. In this way, okay, now the repressor protein is going to leave the operator which allows RNA polymerase to bind to the promoter, and then gene expression occurs, and then this produces proteins that are enzymes that break down lactose. Right? So that's a lot of writing. Let's show it in the picture. So default setting, okay, default setting, the operon is off, the repressor protein is active, no transcription occurs. However, what happens when you have lactose? When you have lactose, that lactose is going to fit into this active repressor protein. So here's our lactose right here. It's going to fit, let me try to draw it, into the active repressor protein, causing the active repressor protein to become inactivated. There's the lactose there. Okay, so now we have our inactive repressor protein. And here's our inducer. Here's our lactose that inactivated our repressor protein. So if the repressor protein isn't there anymore, that means that RNA polymerase can bind. It can move down this, right, because the repressor protein has been removed. And you can have transcription of the mRNA. You can have translation of the proteins. And these proteins, these enzymes that are made, can take in the lactose. And like I said, remember lactose is a disaccharide. Can take in the lactose, right, and break it down into glucose, which is a monosaccharide, and galactose, which is a monosaccharide. Okay? So this is an example of an inducible operon. It's off, this repressor protein is active, this operon is off when there's no lactose. When lactose is present, the repressor protein becomes inactivated so that transcription and translation can occur so we can produce this enzyme to break down lactose. So this is our example of our inducible operon, and this is our example of our repressible operon. Thanks for watching.